to Bio 111. This is section 2. We're going to be looking at DNA structure and sequencing. Alright, so structure of the nucleotides. These are building blocks of DNA and RNA. Alright, they're made up of a sugar, a deoxyribose in DNA, and ribose in RNA, a nitrogenous base, either adenine, thymine, cytosine, or guanine, or in the case of RNA, uracil. Uracil replaces thymine in RNA, and then a phosphate group. All right. Now DNA is made up of nucleotides, either the adenines, guanine, cytosine, and thymine, bound together as a chain. All right, so we have cytosine pairs with guanine, thymine pairs with adenine. And if you notice that thymine and cytosine are pyrimidines, guanine and adenine are purines. And the purines, if you also notice, have two rings. The pyrimidines have one ring. So the purines are actually larger structures than the pyrimidines. And it's important to note that adenine, a purine pairs with thymine, a pyrimidine, and cytosine, a pyrimidine, pairs with guanine, a purine. So big to small, big to small. The reason why this is important is because this pairing allows for the DNA molecule to be a consistent thickness or width throughout its length. If two pyrimidines were to be paired together beside two purines, you'd see kind of a wavy pattern in it. There'd be kind of a bulge. All right. All right. So DNA is a double helix polymer made up of repeating nucleotides, bound covalently, linked by phosphodiester bonds. These run in opposite directions. They are considered to be anti-parallel. Two polymer strands are held together by specific complementary hydrogen bonds between opposing nitrogenous bases. Now I want you to notice something here. We look at the binding between the thymine and adenine in this picture. Right, notice there's only two bonds. The binding between guanine and cytosine are three bonds. There's a stronger bond between guanine and cytosine than there is between thymine and adenine. Right? That's somewhat of an interesting property of these two. In some cases, can be used to help identify which binding we're looking at, either between thymine, adenine, or guanine and cytosine. All right, so RNA. So remember, looking back at the beginning of this chapter, we talked about the central dogma. DNA is transcribed to RNA, and RNA is used to translate DNA. The data is translated from RNA into DNA. RNA is very similar to DNA. However, the sugar is just ribose. There's a phosphate group, and then we have our nitrogenous bases, adenine, guanine, cytosine, but instead of thymine, we have uracil. All right. Now, there are three types of RNA that we will talk about. There's mRNA, which stands for messenger RNA. Messenger RNA is the molecule that is transcribed from DNA and will be moved to the ribosome. All right, now in the ribosome, we have a structural RNA called rRNA, ribosomal RNA. Now this RNA functions a lot like a protein and a lot like an enzyme. It is structural, and it acts as a kind of a molecular machine. All right, so it doesn't really have data in it like the messenger RNA does. And then our third type we're talking about here is the tRNA or transfer RNA. And this is also sort of a machine um, function for RNA. What it does is it goes and it has a particular grouping of 3 nitrogenous base pairs, nucleotide pairs. We call that a codon. And it will only bind a particular amino acid based off of those 3 base pairs that it's got. And so what this allows during the translation from RNA to DNA that occurs in the ribosome, it allows for a specific amino acid to be paired to a specific DNA or RNA sequence. All right, so these 3 are very important for the production of protein. Now, we talked about Chargaff's rule, where he found that thymine and adenine and guanine and cytosine are found in equal amounts with each other. So guanine is found in the same amount as cytosine, and adenine is found in the same percentage as thymine in DNA, that is. Now, a scientist by the name of Rosalind Franklin determined that DNA had a helix function. Now you're looking in this B picture here to the right. That is a plate from a X-ray crystallography study. So X-ray crystallography, you can determine, using this method, you can determine crystalline structure by crystallizing a molecule, allowing it to dehydrate and form crystals, and then shining a beam of X-rays through it, and you kind of get what you call diffraction patterns out of it. That's what you see in this picture, and these diffraction patterns, they're kind of like shadows against the wall. You can use them to figure out what the X-rays pass through to make those patterns. And in this case, this pattern is produced by a helix structure. And so they knew that DNA, Franklin knew that DNA was a helix structure. And then we have these 2 people here, Watson and Crick. They're famous for unraveling the structure of the DNA molecule. Now, they used the information from Chargaff and from Franklin to build a 3D model of DNA in which they showed that they had a helical molecule, a double helix made up of base pairs, thymine and adenine, guanine and cytosine, bonded together, right? And that the 2 sides were complementary, meaning that, this is important, meaning that the same data is found in the left side and the right side. So if you take this double-stranded DNA, both strands have the same data in them. And why is that important? Because when we go to copy DNA, we need to have the ability to have 2 templates. Right? So we cut it in half, start splitting it in half. We need to be able to build up the 2 sides again. So they have to have the same data. Now we know that DNA contains all this information that's needed to essentially build living structures. How do we read it? Now, the cells read it, obviously. They're able to take it from DNA to RNA to protein and use it. But how do we read it? And a scientist by the name of Frederick Sanger developed a method to read DNA. In other words, to sequence it. We call it sequencing because we're putting the base pairs in order. All right, so in DNA, the base pairs, adenine, guanine, I keep saying these over and over again because they're important. The adenine, guanine, thymine, cytosine, we're able to put those in sequence, in the proper order. And it is that order, it is that sequence that has the data. If you want to think about DNA as being a sentence or a book, then the base pairs are the letters in the words in that book, and the genes are the words. All right, so we want to be able to read it. We need to be able to know what letters, what sequence these letters are in, what order these letters are in. Sanger sequencing allowed us to do this, and the way it allowed us to do it is allowing DNA replication to occur. So DNA replication is where we're making more DNA to occur up into a point, right? So he had 4 tubes. He filled 4 tubes with each one with all of the needed nucleotides, G, A, T, C, and then he put inside of basically a kill sequence, right? A D-deoxynucleotide. Now, these are particular nucleotides that he utilized that once they are incorporated into the DNA, that DNA strand ceases to extend any further. And so, when you find, what you basically do is you let these run to completion. You allow DNA to be sequenced in each of these tubes. And then, at the end of the day, when you've done this, you can go back and you can look at the lengths of the DNA. And you know, okay, so in this tube I have a termination nucleotide for thymine. It extends 100 base pairs, 50 base pairs, 2,000 base pairs, and we know at those individual places where it stopped that it has a thymine base. And we do that for each one. And we're able to determine the sequence of small strands of DNA using this method. If we want to sequence an entire genome, we've got to do it over and over and over again. It can be very expensive. Now, the Sanger sequencing read these lengths of these DNA by electrophoresis. Now, they do it a little bit different than that when it's automated. But you've probably seen pictures of gel electrophoresis in CSI shows. What's basically going on is you're sorting DNA by size. So let's say that you want to know if 2 people are related. Right? What you might do is you take a sample of their DNA and you chop it up. Right? Now, the tools we use to chop up DNA in specific places. Now, that results in having chunks of DNA of specific sizes and lengths based on whether or not you had these places for the DNA to be cut. 2 people who are not related will not have the same places that allow it to be cut, and will therefore have different sizes of DNA, different chunk sizes of DNA after it's been chopped up. You can show that 2 people are related, or maybe the DNA came from the same person, in case of maybe a criminal investigation, by running the DNA, the chunks of DNA, in an electrophoresis. What this does, you take the DNA, you put it inside this gel material. This gel material is a matrix. It's basically a filter. These shorter pieces have no problem going through this matrix. The longer pieces get caught up. And so what you'll find is that the really small pieces of DNA are at the very top of this, and the really heavy pieces, the big pieces that get caught up in the matrix, get caught up in this gel, are the bottom. And this allows us to sort based off of size of DNA. And you can use this as a rough way to determine if two people are the same the DNA came from the same person, or if DNA is if two people are related. It's also commonly used to determine if a piece of DNA is actually present in a sample. All right, now we talked a lot about the difference between eukaryotes and prokaryotes in earlier chapters. We're going to just talk a little bit about their DNA, their genomes. Now, prokaryotes, they're simple. We talked about this. They're simple. They have these circular chromosomes, if you want to call them that. They're loops of DNA. But there's a problem. When you look at the length of this DNA inside of this tiny little bacteria, they're about the size of a millimeter. If you stretch out the DNA, it's about the size of a millimeter. But these bacteria are tiny. They're smaller than our cells, significantly smaller than our cells. If you remember, it's believed that the mitochondria in our cells actually were some prokaryote that got eaten by one of our ancestors. So these are very small. So how does it get this DNA, this long length of DNA, inside of such a small package and still have room to do other things inside of it? And what it does is something called supercoiling. So the DNA gets coiled up, and when it gets coiled up, it begins to coil up on top of itself. It gets to become really compacted. If you've ever seen a home phone, a landline that has a cord, you stretch it out a lot, and it begins to coil up. The phone line is already coiled, but then it begins to coil up on itself. And you might have a 10-foot long cord that gets coiled up into a foot hanging from the telephone. That's sort of the same idea that happens inside of these cells. Now, our cells, eukaryotes, we have an even longer length of DNA, and so we require significant more compacting and much more complex use of much more complex packing mechanism. And so the way our DNA is packed is that our DNA is wrapped around these spools called histone molecules. These are proteins that bind to the DNA and wrap it around itself, wrap the DNA around the histone molecule. That allows it to become more compact. And those histone molecules then form larger coils, which then form more complex coils, and eventually they can be condensed further into these chromosomes that we saw when we looked at metaphase during the cell cycle. By this means, we're able to have these huge lengths of molecules inside of our cells, and be able to use them. Now, remember, these are molecules. They're long. The longer you have a molecule, the more likely it's going to break. So this compacting actually preserves the molecule. Now, the parts that are wrapped around the histones sometimes have lower access to transcription, so that can affect whether or not that gene that's wrapped around the histone is actually even used or not. And there's something that we've discovered in the last 10 years, 15 years, called epigenetics. And epigenetics, we have found, relies on the activation or the deactivation of some of these histone groups. This histone protein, how well it binds the DNA and whether it will let the DNA go and allow it to be accessible to transcription. Epigenetics is it's called epi because it sits on top of the genes theoretically or kind of virtually, and it it's a um, it's a phenomenon where different sections of the DNA are activated or deactivated, that is, allowed to be transcribed or not allowed to be transcribed, that is actually controlled by environment, which is interesting. We didn't think that environment had a huge impact on the actual expression of DNA, that is, the creation, whether or not a protein or a gene would be expressed, would yield, would be transcribed and translated into a protein. We did not know that that was environmentally impacted or any mechanisms that could allow for it. Epigenetics is that. We'll talk about that in later chapters. All right, so that's it for section 2. Come back to the next video for section